What's up, guys? This is a beautiful whiteboard right now, and we're going to go through a lot of stuff about the blood, the life force. So let's get rocking and rolling. So these are the things we're going to go through today. I labeled them in this center section, and I'm going to refer to these other sections periodically. So first off, we're going to talk about some fun facts. They'll be great. We're going to talk about the blood composition, so what it's made of, and then also how it is formed. I'm just going to briefly describe that. Third, we're going to talk about hemoglobin, its functions, why it helps us, and that's going to bleed into, see what I did there? Bleed into carbonic anhydrase and what it's going to do both at the body cells and the lung tissue. Finally, we've got blood types over here. I'll walk you through that. And then finally, I'll very briefly describe the heart pathway, which is what we finished up with on Thursday of this week. So let's get rolling. First off, fun facts. In 1 milliliter of blood, there's approximately 2 to 6 million red blood cells. In 1 milliliter, that's like this much. That's a lot of blood cells, right? So it's one of the most abundant cells of our body, our red blood cells. Another fun fact is it's about 7 to 8% of your body weight is blood, and you contain about 4 to 6 liters of blood at any given second. It's also one of the, master regulators of your body temperature. So the blood will get warm and cold in different areas, and then it'll flow throughout the body and kind of maintain that same temperature. So it's a big temperature regulator in your body as well. If you were to stretch out your blood vessels end to end, they would stretch around the earth 2.5 times, approximately 50,000 miles. That's incredible. So those are some blood fun facts. I hope you enjoyed. Now let's talk about the composition. If you were to centrifuge out, so spin out some blood, it would separate into 3 distinct parts. We've got the plasma section, we've got the buffy coat, which is negligible, it's super small percentage, less than 1%, and then we've got the formed elements, will actually be the cells of the body. So when we're talking about plasma, plasma contains mostly water, about 92% water, so of that 55 is 92% water. 7% will be proteins, I'll walk through what those do. And then also some solutes. So some solute examples would be ions, potassium, sodium, calcium, magnesium. Nutrients, glucose, maybe some amino acids. Gases would be some things like carbon dioxide and oxygen. And then waste products, maybe things like uric acid, urea, maybe some lactic acid, those types of waste products that will eventually get excreted out of the body. Now let's talk about these proteins. Albumins. Albumins are great fat carrier proteins. These are the carrier proteins that we talked about in the previous section, where whenever we have a lipid-soluble hormone, we have to have a protein carrier. Well, albumins and globulins are big factors in that. So these proteins will basically gobble up, in a way, those lipid hormones or peptides, whatever they are, and carry them nicely throughout the bloodstream. I also talked about albumin as a primary regulator of osmotic pressure in the blood capillaries. So basically at the capillary level, actually I can show you right here. So this is a blood capillary. A lot of fluid will leak out into this interstitial space. Okay, so as this blood flow is coming, there's going to be fluids and nutrients basically diffusing across the blood vessel, going to different body tissues, which is good. But if all of the fluid escaped into the interstitial fluid, well then there'd be no fluid in the blood and your blood pressure would decrease significantly. So what you have inside of your vessels are proteins called albumins, because they end in IN, all of these, well, most of them. And what those albumins will do is it basically attracts a lot of the water back into the blood vessel, into the venous end that will come back to the heart. So the albumins are very important at bringing fluid back into the venous end of the blood vessels after it's delivered its ions and tissues, or ions, fluids, and nutrients to the tissues of the body, reclaiming it into the blood vessel. So I showed you that in class. Hopefully that was a beneficial example. Globulins will do the same thing. I mentioned that they're going to be some carrier proteins for lipid hormones. And then for fibrinogen, that is one of the initiators of blood clotting. So fibrinogen will be secreted into the areas that are cut, like vessels that just got cut. And the fibrinogen will basically be activated, turn into fibrin, and eventually make this wonderful protein net that basically secures that blood clot and prevents more bleeding. So that's the biggest player, and one of the biggest protein players in blood clotting, which we'll talk about briefly later on. So as you can see, 7% were the proteins, mostly water, and then a little bit of solute, about 2%. Great. Let's move to the buffy coat. Buffy coat, I would consider it's part of the formed elements because formed elements are the cells. Okay, so these are going to be your white blood cells and your platelets. Platelets and fibrinogen and a lot of other clotting proteins deal with blood clotting. So platelets will initially clot that hole. So first off, let's just walk through a blood clot. You accidentally cut yourself, right? You're bleeding. What the blood vessels do that got severed will vasospasm. So it's called a vascular spasm where those vessels basically close up, they constrict, so less blood can flow out. Okay, we're going to diminish the bleeding. Then platelets will aggregate around that area, basically forming a plug. And the plug will be held in place by proteins like fibrinogen, fibrin, thromboxane, I always forget that one, and a bunch of other proteins will basically make this beautiful net around that blood clot so that you don't bleed anymore. Okay, so that's a big proponent of blood clotting. White blood cells are WBCs. We got neutrophils, lymphocytes, neutrophils, lymphocytes, monocytes, eosinophils, and basophils. So the things that end in phil, neutrophil, eosinophil, and basophil, those are your granulocytes. These two, lymphocytes and monocytes, will be your agranulocytes, so they're not going to have as many granules if you look at them under a microscope, okay? They're going to have more defined nuclei. Okay, I remember that by never let monkeys eat bananas. Okay, it's just an easy analogy to remember. How much? Because this is like, there's more neutrophils than basophils usually, and we saw that in the complete blood count that we used in class. There were more of these than there were of those. Awesome, and those are primarily used for, obviously, fighting off infections. Depending on what type of infection it will be, there will be different increases in one cell type. It just depends on what it is. Okay, let's move on. So that was blood composition formation. Oh, I forgot my red blood cells. Red blood cells, they're going to be the primary nutrient transporters, specifically oxygen. Okay, and also carbon dioxide is going to help with that, which we'll show in a second. Hemoglobin. Okay, let's talk about that. Hemoglobin. Heme, okay, usually means blood, okay, like hematocrit, right? With hemoglobin, the heme group contains an iron, okay? There's an iron core. And what that iron core loves to do is hold on to oxygen. So the reason your blood is red is because when oxygen is added to iron, it rusts, right? And rust looks slightly red. So the reason your blood is red is when iron binds the oxygen, which is good because the hemoglobin is on this red blood cell, so I drew it here, this little hemoglobin, and it's basically going to grab that oxygen from the lungs, right? It's going to grab the oxygen that you breathed in, and then carry it, and then drop it off at the body tissues. Very important thing that the blood does. However, there's an issue that we talked about in class. When carbon monoxide poisoning occurs, carbon monoxide loves to hang out on the hemoglobin. So it basically blocks the oxygen on that on the iron from getting onto the iron. So now you are hypoxic. You lose amount of oxygen in your body. And the way you treat that is you give them basically 100% oxygen as a mask, and you put them in a hyperbaric chamber, so a high pressure chamber where it's basically going to force that oxygen into the body, forcing the carbon monoxide off of the hemoglobin, and then resaturate it with oxygen to get oxygen to your tissues. Because you need that for cell respiration, to make ATP, to keep your body alive, right? So, great points with hemoglobin and with oxygen binding to that. So, let's move on to carbonic anhydrase, because hemoglobin doesn't just carry oxygen, but it also helps regulate the blood pH, and I'll show you how. So, over here, we've got two different sections. So here, if you can see my marker, we've got a respiring cell that's generating a lot of ATP, water, and carbon dioxide. ATP is good, water's fine, carbon dioxide can be toxic. So carbon dioxide has to be released by the cell into the interstitial fluid, into the capillaries, which can only fit one blood cell at a time. So the blood cell notices, hey, there's carbon dioxide popping in here, but carbon dioxide is a nonpolar molecule. That doesn't mix well with your blood, which is mostly water, because water is hydrophilic, carbon dioxide is hydrophobic. So we've got to fix this problem by turning carbon dioxide into an ion, because ions, charged atoms, or elements, or molecules, can flow well in water. So how do we do that? Well, we've got water floating around, and we've got our carbon dioxide coming from our respiring tissues. An enzyme called carbonic anhydrase will transform it into, first off, carbonic acid, which I didn't write, which will then turn into a bicarbonate ion and a hydrogen ion. Okay, so the goal was to turn carbon dioxide into its ionized form, which it just basically did through this enzyme, synthesizing it into carbonic acid, which then dissociated into hydrogen ions and bicarbonate ions. Bicarbonate ions are fine. They don't usually bother things too much. However, hydrogen ions, we know, make your blood more acidic. So, if you have too much carbon dioxide getting transformed into hydrogen ions, well, now your blood pH is going to drop like a rock. So, what happens? Your hemoglobin, miraculously, will act as a molecular Swiffer, and it will Swiffer up all of those pesky dust mites, which are hydrogen ions. So the hydrogen ions are like the dust, the hemoglobin's sweeping it up, so it resists that drop in pH and maintains a good, healthy pH range in your body. So it's a very important tool that your body uses, this carbonic anhydrase and hemoglobin scrubber, to maintain your body's, your blood specifically, pH. Okay, so that's incredibly important. Next one, let's talk a little bit about blood type. Oh wait, no, what am I doing? That was at the respiring cell, right? So it was making CO2, transforms into its ion, hydrogen ions get scrubbed up. But then, we need to get the carbon dioxide out, because eventually we're going to be chock full of hydrogen ions on our hemoglobin. So we got to get the carbon dioxide out eventually, right? Let's go to the lungs. If you notice, this is the reverse process. I showed you guys a diagram of this, but what will happen is bicarbonate and the hydrogen ion will be released from the hemoglobin, combine into carbonic acid, and then carbonic anhydrase will turn it back into carbon dioxide and water. So that way, it diffuses out of the blood capillary, the pulmonary capillary, and goes into the lungs to get expelled. Okay? So the reason we changed carbon dioxide in the first place was to make it an ion. Okay, so it transfers in the blood well, but then once it gets back to the lungs, we need to reverse the process, turn it back into a gas, essentially, so that it can get out through the lungs and out of your body by breathing out. Okay, complex concept. I think you guys understood it well in class. Let's go to blood types, and then we'll talk briefly about the heart pathway. We talked about blood types at length, okay? And a lot of this just comes down to studying, okay? So I posted the chart on our learning management system to show which ones can donate, which ones can receive certain types of blood. Use that to test yourself, but by no means memorize it. Okay, so I'm going to walk through it very briefly. On your blood cells, there will potentially, depending on your blood type, be antigens. Antigens are found on the cell membrane. They're basically like an identifier, a flag of your cell that says, hey, I am this type. There are 3 antigens, okay? One antigen is your A antigen. So if you have type A blood, your blood contains these black squares, hypothetically. If you have antigen B, type B blood, you have a pink triangle. If you're antigen plus, you are plus, so like B positive, B plus, or A positive, A plus, AB positive, whatever. So, those are the antigens that are present on the outside of your cells. What blood type would this be? Well, it has a positive antigen, so it's got to be a positive, but it doesn't have A or B. So the light's kind of weird. So if it doesn't have A or B, but it has the positive, this would be called O positive. Okay? O meaning no A or B, but it does have a positive antigen on the outside. Okay. Let's go to the second type, and then we'll talk about which ones they donate and receive from. Second type, you see, has pink triangles, black squares, but no pluses. This would be AB negative. Negative because there's no positive antigen. Okay? So this guy would be AB negative. Okay. O positive. Okay, O positive. First off, who can they donate their blood to? Well, they can always donate it to themselves. So if there's another person with O positive blood, they can donate it to O positive. Okay? They can also donate it to any other blood type that also contains their antigen, in this case. So if they are A positive, think about this, they'll have the A antigen. They'll also have the positive antigen. This one only has the positive antigen, so it won't bother the A. The A is like flying its flag, and this O positive blood will come along, and it'll just be like, you know what, I've got a positive one as well, so I have the positive antigen. Come on in, it'll work just fine. So they can also donate to A positive, which also holds true that they can go to B positive as well. They can also donate to AB positive too. So we can also go to AB positive. Hopefully that makes sense. I went over it a lot in class. So they can donate to those, but they can only receive it from two types themselves. So they can receive from O positive, and they can also receive from O negative. O negative has no antigens, so it will be the universal donor. It's basically like Switzerland. It'll play nice with everybody, and it won't bother anybody. Okay? So O negative is the universal donor. Now let's go to AB negative blood. This one's a little tricky. They can only donate to two, AB negative and AB positive. Okay? So AB negative, AB positive. If it donated, for example, to B negative, okay, you'll say, well, Mr. Jackson, it's got a B, so shouldn't it be happy? Well, yes, but it also has an A. And A doesn't like B, right? So if it were to, sorry, I don't know what the example I was giving. B negative? Yeah. So it has the B antigen, but also has the A antigen. And A doesn't like messing with B's if it's donating to that. So basically what would happen is you donate the AB negative blood into a B negative person, and the B negative person doesn't like A. And our blood type does have A in it. So it would basically start attacking that A antigen. Okay? Who can they receive from? A lot of people. Okay, so let's think about this. They could receive from O negative, because it matches the negative, and it's the universal donor, so we know that O negative can go to everybody. Can also receive from B negative. You can also receive from A negative. And I missed one, A, B negative. Okay, so itself. So basically anything that is negative. The issue with this, what if it got it from O positive? Well, that O positive would have a positive antigen that doesn't match any of these, so he would freak out and be like, I don't know what this is, I'm going to kill it. Okay? So it can't receive anything positive. Now, if it was AB positive, hypothetically, that is the universal recipient, because it can receive O negative and positive, A negative and positive, AB negative and positive, B negative and positive. It can receive everything because it has all the antigens. Therefore, it doesn't make any antibodies against anything. So that would be the universal recipient. Again, if you need to watch other videos to try to walk through that yourself, please do, and just study it up as best you can. All right, last thing, we're going to talk about the heart pathway. So I'm just going to erase a little. Actually, I can do it right here. The only thing we went through with the heart pathway is we drew a heart. We cut it into 4 sections. Okay, this would be the left if you're looking at somebody. This would be the right. The bottom sections are the ventricles. So this would be the right ventricle. This would be the left, or I'll just say left ventricle. The right top and the left top are both called atria. So this would be the right atria. This would be the left atria, or atrium, I guess. Atrium is singular, atria is plural. The left ventricle will pump blood to the body. It is oxygenated, and it will feed the body with oxygen-rich blood. The right ventricle will pump blood to the lungs. The right ventricle will pump the blood to the lungs. This is deoxygenated blood, so it's going to the lungs to basically pick up more oxygen. Okay? The atria will contract synchronously and push blood into the ventricles. So basically it's this big circle, so it's going into atria to ventricle, and then this side goes to the body, head to toe. This right ventricle goes to the lungs. Okay? The right ventricle will be a little smaller because it's just pumping the lungs. So my heart's here, my lungs are here. It doesn't have very far to pump, so it doesn't need to be too big. The left ventricle, though, the left side, is a lot bigger because it's pumping all the way to the top of your head to the bottom of your toes. So it's got to be really forceful and contract nicely. Okay, hope this was helpful. Watch it again if you need to. Again, this is Mr. Jackson, Organized Biology YouTube. I hope this helped.